Well, welcome everyone. I'm Joanne Geer. I am the Executive Director of the Biopharma Research Council. We are an association for scientists throughout the biomedical research process. We are interested in bringing people together across all the different silos of biomedical research, which include industry, academia, nonprofit, government, supplier labs, and their teams. And we do a number of webinars throughout the course of the year, as well as some physical events. I love this slide. This is from a conference that we did last summer called D2D, Data to Drugs and Diagnostics. I think everyone struggles continually with how do we collaborate and how do we save IP. So it's a really interesting context to look at the growing cohort of IT people who can really help scientists and scientists who can really get a lot of help if the parties learn how to talk to each other. So. In May, we'll be in Boston. Merck Research Labs actually invited us to do this program, Advancing Drug Discovery Through Genome Engineering. It's largely about CRISPR, but also some other technologies and models, various models. One of the reasons that they asked us to do this conference is that they are looking for partners to work with as they go forward to try to develop therapies using these technologies. And this is just to share a few of the different organizations and institutes that have supported us through providing speakers, providing sponsorship. We have a really wonderful community of about 8,000 researchers that we work with on a regular basis. I want to especially thank InTech, which is the open source publishing company that's sponsoring this series, IES Diagnostics, the NIH, you'll be hearing from Howard Young in a moment, and Planet Connect, which is our primary and founding sponsor that makes almost all of our programs possible. At any time, you can add a question to the box on the side. We'll be taking questions at the end of the presentation and have a nice long time for discussion. We're going to hear from Rob Knight today. I'll be reading you his bio in a moment. Unfortunately, Dr. Kuo isn't able to be with us today, but Howard Young is on the team, and he'll be joining us for the conversation. Rob Knight is recently the recipient of the Vilcek Prize for Creative Promise in Biomedical Science. We're very happy to have him here with us. He's done a couple of other webinars for us, which are available on our website as recordings. He is a professor of pediatrics and computer science and engineering and director of the Microbiome Initiative at UC San Diego. He is co-founder of the Earth Microbiome Project and the American Gut Project, and co-founder and CSO of Biota Inc. He has co-authored more than 300 peer-reviewed research articles and has been cited more than 41,000 times with an H-index of 86, according to Google Scholar. His research includes the development of highly multiplexed techniques for reading out the human microbiome and the microbial communities in environmental samples, software for analyzing microbial communities, including the popular QChemie and Uneprack packages, and high-throughput genome analysis. He has applied these tools to uncover links between microbiomes and obesity, malnutrition, inflammatory bowel disease, and diabetes. He is senior editor at ISMEJ on the editorial board of Cell Host and Microbe, Genome Research, Genome Biology, and several other journals. I'm going to briefly introduce Dr. Howard Young, who is the Deputy Chief of the National Cancer Institute Laboratory of Experimental Immunology. Most recently, his laboratory has developed novel mouse models of lupus and aplastic anemia, and is collaborating with a laboratory in Malaysia on the development of probiotic microorganisms as carriers for vaccine delivery systems. So here we are, and we're ready to go. Dr. Knight.
Okay, thanks Joanne for that very kind introduction. Thank you all for coming along. I know you had a wonderful seminar from Paddy Sabeti yesterday, and I'm glad you're ready for more cool stuff about the microbiome. What I talked about last time was more of a general introduction to how we can assess the microbiome overall and how we can look at causality. And what I'm going to talk about today is a more specialized topic, looking at spatially explicit maps. In other words, how can finding the location of microbes and the metabolites they produce tell us a lot more than we can find out by other means. So the key question here is, how can we map our microbes? And we really take a leaf from macroecology, where when you look around different parts of the world, you see indicator organisms and landscapes that immediately identify a scene as coming from one location or another location or another location. With microbes, it's kind of similar, although I'll be honest, if you've squinted down a microscope in introductory biology, you know that you're facing that problem that a lot of the microbes look more or less the same. So instead, what we do is we tend to their DNA. And for example, in the Human Microbiome Project, together with a team of about 400 researchers around the country and around the world, we collected about 4.5 terabases of data, so about 4.5 trillion A's, T's, G's, and C's from 250 healthy people, sampled at up to 18 sites on the body. And so that's an impressive amount of data, but the drawback is that the data look like this, and you're faced with the challenge of interpreting it, right? It's pretty hard to see who lives where in the environment now when you just get back the raw sequencing results. And I'm guessing you can't even pick out that this happens to be an oral sample, for instance. This is the first file of data from the Human Microbiome Project, looking at the first oral sample from the first time point from the first individual, but there's no way you can tell that by just squinting at the sequence files alone. So the focus of my lab for the last decade has really been on developing computational methods, which you'll soon learn a little about and even how to pronounce them. The first key idea is this idea that we can exploit evolution to compare different communities to one another. So this is this technique called Uneprack that Kathy Lozapone and I introduced back in 2005. And the intuition here is that if you have two unrelated communities, like over on the right-hand side, so these are phylogenetic trees where the squares represent modern species, and then the internal nodes represent inferred common ancestors over evolutionary time. So if you have two unrelated communities, like in the tree on the right, all of the branch length is going to lead only to members of the red community or only to members of the blue community, but never to both, because you can't have an organism moving from red to blue and surviving and reproducing and contributing new branch length. Now, in contrast, if you have two identical communities, like over on the left-hand panel, everything you find in the red community, you also find in the blue community, as long as your sampling is complete. So all of the branch length on the tree is shared, shown here in purple, and we define that distance as 0. Then for any actual tree, you have some shared branches shown in purple, some unique ones shown in red or blue, and the fraction of the unique branches, or Uneprack distance, is going to be some number between 0 and 1. So what's so great about that is we can use Darwin's insight that there is one phylogenetic tree that relates all organisms to take microbial communities from any environment and put them on the same tree. So here we have one tree with red, yellow, and blue communities. So we can compare red to yellow and red to blue and yellow to blue using the technique I just showed you, and then summarize all that information in a distance matrix. And then once we have that distance matrix, we can do things like principal coordinates analysis and hierarchical clustering to figure out figure out a layout of how those communities relate to one another at the whole community level. And so we integrated this into an overall pipeline where what we do is initially we tag the 16S ribosomal RNA gene from hundreds of different samples using DNA barcodes that we introduce. And so the reason we use 16S is it's a very good phylogenetic marker to give you a readout of what organisms are in a community. Then originally we sequenced on the 454 instrument, although more recently we've moved to Illumina. And then because each sample has a barcode, we can tell, because each sequence has a barcode that we introduced during PCR, we can tell what sample it came from. Then we can build multiple sequence alignments, build phylogenetic trees that color the samples on the branches, and then use those trees to color the samples. For example, you might expect different clusters of healthy versus disease samples. And all this is put together in a pipeline called QIIME, which stands for Quantitative Insights into Microbial Ecology, and basically takes the raw data off the sequencing instrument and goes all the way to visualizations and statistics that you can use in a publication. So, so it's free, it's open source. What we use in our lab is exactly the same as what's on GitHub that you can download publicly, and it'll run anywhere from your laptop to supercomputers like the Hopper instrument at NERSC with 140,000 cores to the Amazon EC2 cloud. And when we apply it to the HMP dataset, what we get is this map that looks like this. So each point on this graph represents a microbial community. We're looking at the principal coordinates reduction of Uneprack distances. So for example, this complex oral biofilm, all of those microbes are just going to be condensed to one point on the map. And so 2 points that are closer together have more similar communities. 2 points that are far away have more distant communities. And you can see some patterns emerging automatically. So you see a big cluster for the oral microbes, a big cluster for the skin microbes in blue, another one for the vaginal microbes in pink, and then the stool is down at the bottom in brown there. So what this implies is that two samples, even from the same body, can be very different in composition. So for instance, if I highlight one person's oral community and their fecal community, you can see that they're very far apart on that map. But how far apart are they really? Well, one of the great things about being able to use phylogenetics is all the organisms are related, as I mentioned, so we can use any kind of environment as a reference standard. And so if we say that that oral community is kind of like a coral reef, in that you have these complex carbonate structures, you have all these complicated biofilms that may be your dentist pestis you about and so forth, then the microbes in the mouth are as far away from the microbes in the gut of the same person as the microbes on this reef are from the microbes in this prairie soil. So they're essentially non-overlapping communities. And so that means that a few feet along the length of the human body makes more of a difference to microbial communities than hundreds of miles across the Earth's surface. Now, all the stool samples look more or less like they're focused down on the same area in brown here, but that doesn't mean they're the same. So if I highlight a second person's stool, it's as far away from the first person's as the microbes in this prairie soil are from the microbes in this forest soil. So in other words, it's about 10% overlap between those two communities for two arbitrarily chosen people. And that's pretty remarkable when you think about it, because we're all 99.99% the same in terms of our human DNA, but we might only be 10% the same in terms of our microbial taxon inventory. And remember that our microbial genes form far more of the genes in the system that makes up our body than our own genes do. We only have about 20,000 genes, whereas the size of the microbial gene catalogue has been estimated from 2 to 20 million. So this might lead you to wonder how stable is the microbiome, because in this abstract space I'm showing you a lot of differences between different people. And so Amanda and I, my partner Amanda and I, to address this question, sampled ourselves every single day for a period of 6 months. So I'm showing you this in an earlier and somewhat simpler data frame from the Costello et al. 2009 data, but the colours are the same, with the mouth in green and the skin in blue and the feces in brown. And each frame in this is one day in the life of our microbiomes over this 6-month period. And I'll start that going, and you can see immediately how variable the skin is, even within a person, how static the mouth is, and how the gut is in between the two. And you can also imagine how in this kind of abstract space, it could be incredibly useful for tracking people, not just in this kind of case where nothing in particular is going on, but in cases where you have a therapeutic intervention, or in cases where you have a process of development, or that kind of thing. And you can also see we maintain our separate microbial identities during this period. So I'll just spin that around for you. Despite the fact that we cohabit and have all kinds of opportunities to exchange microbes with one another, you can see that we maintain our separate microbial communities throughout this 6-month span. So this might lead you to wonder, where do our microbes come from in the first place? And if you have dogs or kids as I do, you probably have some dark suspicions about that, all of which turn out to be true, by the way. So we can match you up to your dog with pretty high accuracy just by the microbes you share. But in all seriousness, how we're born has a tremendous role in shaping our first microbiomes. If we're born vaginally, all our microbes look like our mother's vaginal samples. So here I'm showing you work we did with Maria Gloria Dominguez, now at NYU. And so what we're looking at is mothers an hour before they gave birth, and then babies within 20 minutes of birth. And so the babies that are born vaginally, all the sites from all over their body in pink look like the mother's vaginal samples in red. Whereas those that were delivered by C-section in light blue, all of their microbes look like their mother's skin community in dark blue. So that might lead you to wonder, where do you go after that? And this is work we did with Ruth Lay's lab, looking at one infant fecal sample over the period of 2.5 years of life, basically until the kid was out of diapers, after which which it's a lot harder to collect those fecal samples. And so we're starting with the meconium, which looks more or less like a vaginal sample. And the question is, over those 2.5 years of development, how does the infant's fecal sample come to resemble the adult samples? Is it a complete progression or only part way? Is it smooth, or is there a lot of back and forth? So when I show you that, and remember each frame in this is 1 week, what you can see is that sometimes there's tremendous variability 1 week to the next, sometimes there's not much variability. Remember that what matters is how far apart two successive points are. And so what this means is that the change in the same kid 1 week to the next can be larger than the difference between any two samples we looked at in the entire human microbiome project. So coming up here is fascinating. He's about to get antibiotics for an ear infection. And what you can see is this tremendous regression of the developing microbiome, followed by a rapid recovery. That went by pretty fast, so I'm just going to rewind it for you and play it again. What we see is that when we give the kid antibiotics, there's a tremendous regression in the microbiome, undoing months of development, followed in this case, but not in all cases, by a relatively rapid recovery. And then by the time he gets to 2.5 years, he looks more or less like he has an adult fecal community. We can also do this sort of thing cross-sectionally. So this is work we did with Jeff Gordon's lab and a number of others, looking at development over the first 3 years of life. So what you can see is in an African population in red, a South American population in green, and in the US population in blue, the approach to the adult state, shown by the y-axis, where lower means more like the adults, that approach to the adult state happens very consistently over the first 3 years of life, although there's still a lot of questions about the impact that those early life changes have on the microbiome, and in particular whether drugs you apply early on have a lasting consequence. However, when I show you a principal coordinates plot of the same data, what you can see in the panel in the middle is that the main thing that's going on is age structuring the microbiome from the youngest kids in red to the oldest adults in blue. So the rate of approach is the same, but where you wind up is totally different. And in the right panel, you can see that the US population is totally different from the two non-Western populations. And this is terrifying from the perspective of drug development, because remember that even large-scale projects like the Human Microbiome Project and like MetaHit are only sampling healthy Western adults for the most part. So you're only covering that much of the possible space of configurations of microbial communities we now know about. As soon as you go into kids and as soon as you go into non-Western populations, you find totally different configurations of the microbiome, which might be doing things like metabolizing drugs in completely different ways, affecting safety and efficacy when you try to test the drug in one population and then deploy in another population with a totally different microbiome. So what I've shown you so far has been mostly based around abstract spaces, and so one really important question is how can I do this in a more spatially explicit way that uses the spatial nature of the data? And for this we're going to focus on skin, which it's a lot easier to sample in a spatially detailed way than, for example, your gut. The skin's also intrinsically very interesting because it provides a barrier between you and your environment, and both in terms of the microbes and in terms of the chemicals that are going to be applied. So one of the first forays into this was done by Chris Lauber, at the time a postdoc in my lab, and he was interested in the microbes on the human face, specifically in his own face. This was a collaboration with Noah Ferrer that was sponsored by 454. Of course, with this kind of information, it's really difficult to de-identify the samples, right? Human subjects' concerns can be substantial in a way when you're using spatially explicit data in a way that they're not otherwise. And so anyway, what Chris did is he divided his face up into 50 different patches, and we sequenced the microbes on every patch. And when we do a principal coordinates plot of that, you can kind of see some patterns. So for example, you can see that points 36 and 37 in that blue-green color on the left are very different from the other samples. And what those are the lips, where the microbiome, like you'd expect from the oral samples being different that I showed you before, the oral microbiome is very different from the rest of the face. And there's some clustering by color, and what we're seeing there is primarily that adjacent patches are colored, have adjacent numbers, and so you might expect them to cluster together on the diagram, which they do. But it's kind of difficult to see what's going on overall in this abstract space. What you'd really like to do is you'd really like to be able to paint the data directly onto a map of the face itself and see if that provides additional insight. So Antonio Gonzalez, a very talented postdoc in my lab, figured out how to do exactly that. And so what you can do is you can load any arbitrary picture that you draw. This is a tool called SitePainter that Antonio developed. So you can load in any arbitrary picture, like a map of Chris's face. And if I color it by principal component 1 of the microbial community, you can immediately see a whole lot of left-right symmetry that you just couldn't see in the abstract space. If I show you PC2, what you can see is that there's one patch that's really different from all the others. We think what's going on there is that our subject wears glasses and tends to adjust them from time to time, and the fingertip community is very different from the community on your face. Then if I show you PC3, what we're mostly seeing is the distinction between the lips and the rest of the face. So that's what happens when you relate the abstract coordinates to the spatially explicit map. But the other thing you can do is you can just paint on the abundance of different taxa that maybe you know something about. So when we load that on at the family level, what we can do now is, for example, just look, say, at the Prevotella, which are a classic oral taxon. You can see that there's a whole lot of it on his lips, and then there's a little bit on his cheek, and a little bit in one of his ears. And I'm sure you can come up with your own scenario for how those oral microbes might have got to those two locations, right? And then if I show you, for example, Propionibacterium, which is a classic skin bacterium, which is hiding over here. You can see that Propionibacterium is everywhere on the face, except on the lips where it gets washed out by the oral community, and then on the ears, which stick out from the rest of the face, so they're usually a few degrees colder, and that's restricted for Propionibacterium growth, so they can't live there. So you can really get a lot of information by looking at these spatially explicit maps that you can't get from looking at the similarities and differences between communities in an abstract space. So more recently, with Peter Dorastein's lab, what we've been looking at is maps not just over the face, but over the whole human body. So what Peter did is he took two human subjects and sampled them at about 400 locations on the body, and so he ran all the metabolites using two kinds of mass spec, and we did 16S ribosomal RNA sequencing on the bacteria from every patch, so we could see the relationships between the metabolites and the similarities and differences in the microbial community. And when we do the principal coordinates plots, you can kind of see some patterns. So, for example, let's see, I think this is actually reversed because it's the microbes that are much less distinct between the two people and the metabolites that are much more distinct. So, so it should be the microbes on the left where you see a lot of overlap between the two people, then the metabolites on the right where you see the two people separate much more. But again, it's difficult to tell what's really going on in the body from these kinds of abstract displays. So, so what we did is we painted the data directly onto a 3D map of the body. And so what you can see for particular mass peaks is that there are some mass peaks that are globally distributed over the body. There are some that are localized to, say, the armpit or the groin or the toenails. And basically what we're using is we're using blue, a scale of blue to red, to look from the least abundant to the most abundant communities. Metabolites on the body. And we can do exactly the same thing for the microbes. So you can see that Staphylococcus, like you would expect, is mostly in the moist areas, like, for example, the nose and the feet. Propionibacterium is mostly on the head and shoulders, and Corynebacterium is more broadly distributed over the whole body. However, so one thing that's very interesting about this is we can use a tool called molecular networking that Peter Dorastein introduced together with Nuno Bandeira and Pavel Pevsner here at UCSD, where what you can do is you can relate the spectra to one another based on their similarity, and then you can take your unknown samples and then match them up to spectra from known samples, including skin bacterial cultures in yellow, human skin cultures in green, beauty products in blue, and then chemical reference standards in red. And you can see there's a whole lot of blue on this graph. And fascinatingly, over 90% of the metabolites that we could identify came from beauty products like sunscreen, moisturizer, deodorant, shampoo, and so forth, which is an incredibly high level. And one thing we're just starting to do is to look at the Hadza, the lost hunter-gatherers in East Africa, who have never been exposed to any of these products, to try to understand what a natural skin metabolome would look like before it's had all these chemicals added to it. One thing we can do is we can confirm ideas about particular metabolites, essentially by spiking in particular chemical standards, and then checking that the retention time and the spectra match up to what we expect. And in particular, we can check that metabolic breakdown products have a particular compound, make up the adjacent nodes in the network, and then test hypotheses about what those compounds are, again by matching up the retention times in LC-MS and the fragmentation spectra through MS-MS. So the really cool thing about having a spatial map of all the microbes and all the metabolites is that we can start to infer hypotheses about what metabolites, what microbes are doing what biotransformations. So for example, on the left panel here, you can see that Propionibacterium is primarily confined to the head. Then if you look at something like oleic acid, you can see that there's a lot of it on the head and a lot of it on the hands. So what's fascinating is in the third figure, you can see that the oxidized oleic acid is only on the head where the Propionibacterium is, and not on the hands where it isn't, despite the fact that the oleic acid precursor is in both locations. And then for something like palmitic acid, you can see that its breakdown products, monoalein and monopalmitin, also appear to be found primarily where the Propionibacterium is. So the spatial coincidence leads us to suggest that perhaps the Propionibacterium is doing that transformation. But we can do more than just suggest this stuff. We can actually test those hypotheses rather than just infer them. And so what we can do is essentially we can take the microbes from different parts of the body, and then we can culture them, and then we can feed them the precursor. For example, feeding Propionibacterium acnes triolein, which we think it's going to break down into oleic acid and into oxidized oleic acid, and then we can prove that it actually does that in a way that you don't get if you just run the triolein alone or if you run the Propionibacterium alone. And so this is really exciting because not only can we draw from the spatial colocalization conjectures about what's going on, but we can actually prove that particular microbes are doing that task, and that those microbes are present in some parts of the body but not others, explaining the chemical distributions at different sites in the body. We can also do this sort of thing in the built environment. So this is some work we did with Uneprack, looking at residential kitchens. And essentially what you can do is you can use that site painter tool I showed you before to draw a map of the kitchen. And then what you can do is you can ask where a particular taxa you're interested in, like Campylobacter, Salmonella, Clostridium, Escherichia, and so forth. And the different microbes have different distributions. But what's really interesting is we can also use a technique called source tracking, a Bayesian technique at the whole community level, to ask what are the sources of the microbes in those different locations. And so, for example, when we look at human skin, you can see there's a lot of it on the trash can and on the microwave and on the door handles where people are touching them directly. If you look at the produce signal in green on the right there, you can see that there's a lot of produce signal on the counters and in the fridge. And then in the blue panel at the bottom, you can see that there's a lot of tap water signal, primarily confined around the faucet. And we can do the same thing in bathrooms as well. So in another study with Noah Ferrer's group, looking at bathrooms just down the hall from our lab, which was then in Boulder, you can see that there's a strong skin signal on parts of the bathroom that you touch, like the door handles, the flush handle, the toilet seat. The stool signal, fortunately, is confined mostly to the bathroom stall, but then we see a very clear soil signal both on the floor and on the flush handle. And apparently this is coming from people flushing the toilet with their feet, which I have to admit is a practice that I had never heard of, but apparently it's fairly common in the US. So having these spatially explicit maps lets us pick up all kinds of things about behavior, whether we're doing the maps on people themselves or in the built environments where people live. And we're taking this a step further with our hospital microbiome project, which is led by Jack Gilbert at the University of Chicago, where what we're doing is we're looking at bacterial and fungal diversity over 12,000 samples in the hospital, and then linking it up to over 8 million data points from the built environment, where essentially we can see what the role of temperature and humidity and all of these other factors is, as well as the patients and structuring the communities in that hospital environment. And we think this is going to be incredibly useful ultimately for infection control. And so basically we think the future is going to involve having a microbial profile in your medical records, both for you and potentially for the hospital room that you're dwelling in. So with this excursion into spatially explicit maps, you might be wondering, are we still going to be doing those abstract maps with principal coordinates analysis? And the answer is yes, they can still be very useful in some circumstances. And you can even put yourself on those maps if you're interested. So a couple of years ago, Jeff Leach and I launched this project called American Gut that essentially exploits the decline in the cost of sequencing technology to let people place themselves on this microbial map. Of course, it turns out that not everyone wants to know. These are middle schoolers doing a tour of our lab, learning that we're going to use lasers and robots to look at their poop. But a lot of people have been interested so far. So this is Daniel McDonald, one of the grad students in my lab, who's really been leading the project, which at this point has raised over $1 million. We've shipped out over 9,000 kits, and we've sequenced over 4,500 samples and deposited all the data freely in EBI for any researcher, any educator, anyone at a company to use, including 400 samples from participants in the Personal Genome Project. But we still need to do a lot more of this, especially when we want to start stratifying and asking, say, do antibiotics have the same effect if you're a vegan versus if you're a carnivore? There's all kinds of things that we just don't know yet, and we need more people to find them out to get enough people in each stratified group. And we're finding out all kinds of fascinating things. For example, you might have expected that your microbial diversity would change as you age, which it does, but it also changes depending on how much sleep you get at night. And the findings overall have been very counterintuitive. So what I'm showing you here are power curves, where on the x-axis it's the number of observations you need per group to detect a difference. And on the y-axis is the statistical power of the test. So 0.8 means 80% power to detect. 1 means 100% power to detect. And so you can see that things like inflammatory bowel disease, you don't need many samples to detect the differences. But a factor that has just as big an effect as IBD is the number of different kinds of plants you eat every day. That has a larger effect than antibiotic use or age or BMI or even things like sleep duration and alcohol use, which we can also pick up. The most compelling reason, though, about why you might want to place yourself on these abstract maps is probably the case of Clostridium difficile infection, though. And so this is just to reorient you on the abstract map that we get out of principal coordinates analysis with the different sites of the body defining the different locations. And so what we're going to look at is we're going to look at some C. diff patients, just looking at their stool. And these are people who go to the bathroom several dozen times a day. C. diff is one of the most common hospital-acquired infections, and it kills about 14,000 people a year in the United States alone. So if we look at the stool from some C. diff patients on the same map, this is work with Mike Sadowski and Alex Karutz at the University of Minnesota. What you can see is they have this very profound dysbiosis compared to people with healthy stool. So the microbial community is radically altered. And what's going to happen is that 4 of these people are going to get fecal transplants from one donor, who you can see is in the healthy region of the map. And so, so the question is, what happens after they get this fecal transplant? Are they slowly going to come to resemble the donor? Are they going to stay where they are? How is that going to relate to their health and so forth? So each frame in this is going to be one day in the life of these patients after fecal microbiota transplant. What you can see is essentially immediately they go from the unhealthy region into the healthy region. All of their symptoms of diarrhea clear up almost immediately, and they remain healthy for the several months of follow-up, remaining very close to the donor sample. And so there's a lot of fascinating questions this raises about engraftment. By the way, the last large-scale trial of fecal transplant had to be stopped early because they were comparing FMT to antibiotics, and the FMT was 95% effective, whereas the antibiotics were only about 40% effective. So it was considered unethical to continue withholding FMT from the people who could benefit from it. So the challenge that we face at the moment is to understand for which other diseases besides C. diff that have now been linked to the microbiome, everything from obesity to inflammatory bowel disease to rheumatoid arthritis to colon cancer to atherosclerosis, and even in mouse models, things like autism and depression and multiple sclerosis. Can we do the same kind of mapping task on either in an abstract space or in a real space, where you're essentially trying to figure out trying to figure out not just where you are right now, but where you want to go and what steps you need to take in order to get there. So we really need to develop this kind of microbial GPS that tells you exactly what directions you should go in order to reach a healthy microbiome state. And ultimately, we need to develop this microbial GPS and make it so easy to use that even our children can use it. You could imagine a kind of smart toilet that gives you an instant readout of where you are in the microbiome that's delivered to your smartphone, perhaps with some recommendations about what you should be doing differently or what you should be doing the same, whether particular things are working or not working, and perhaps recommending different kinds of interventions, everything from antibiotics to probiotics to prebiotics to phage therapy, to potentially even things like FMT, and helping you understand which actions you take have particular consequences for where you are on these microbial maps. So with that, I'd like to briefly thank the large number of people involved in this work, many of whom I've mentioned specifically throughout the talk, so I won't reiterate that again. I'd like to thank the large number of people currently or previously in my lab who've contributed to this or to other projects. And finally, I'd like to thank you for joining us today and for your attention. I'd be delighted to answer any questions you have. Thank you.
Well, thank you very much. This is terrific. We do have some questions. I'll start reading them. Our first question is, one theory is that drugs change the trajectory of the microbiome during growth. Are there any efforts to compare with non-human subjects?
Yes, so Marty Blazer has been doing a lot of work on that in mice at NYU, and so what he finds is that antibiotics, especially early life antibiotics, have effects on everything from adiposity to bone density. So it's a very active area of interest in model organisms now. The big question, as always, is going to be which of those conclusions transfer quantitatively to humans. But it does seem that not only does it matter what antibiotic you get and how much of it you get, but additionally when you get it during development.
Thank you. Here's another one. Has anyone speculated on the effect that cooking had on early selection pressure of human gut microbiomes?
Yes, a number of people have speculated on that, including Steve Lee, who's the Dean of Arts and Sciences at Colorado, and we have a joint postdoc, Katie Amato, who's been doing a lot of comparative work looking at primate microbiomes. At this point, it's relatively difficult to pin down, but it's certainly true that you won't find any raw food enthusiasts who are obese, and it's also certainly true that cooking releases a lot of energy from food that we and our microbes are otherwise unable to digest. So it's very likely that cooking has a large effect on our microbiomes and possibly on our ability to derive enough energy to support our enlarged brains, for example. On the other hand, whether it's quantitatively important compared to other things like, for example, changes in physical activity, which has an effect on the microbiome, and other changes in diet, and especially the ratio of carbohydrates to proteins, which has a massive effect on the microbiome, we don't know yet. But it's certainly a very active area of research, both in terms of comparing humans to non-human primates and in terms of comparing human populations around the world who have very different lifestyles and diets, including very different balances of cooked versus raw food.
Thank you. Is there any follow-up on the...
I had a couple of questions for you.
Sure.
Okay. First of all, Rob, do you see this MinION technology beginning to generate more microbiome sequence data in a faster way?
Faster, but useless so far, unfortunately. We hope they'll be able to overcome the problems with it soon.
Okay, and there was one slide that I didn't quite understand the data. You showed both the metabolome and the microbiome on individuals, and you showed where the microbiome was similar, but the metabolome was unique to the individual, and I didn't quite understand how that could occur if the microbiome was similar.
Correct. So the microbiome was similar between the individuals. The metabolome was more distinct across the body. We believe that the reason for that was due to the large contribution of beauty products, which differed between the two individuals. So it was one man and one woman, and as you might expect, despite sharing a household, they did not share any of the cosmetics they applied and so forth. On the other hand, one thing that was surprising about this, and you can read much more detail in our recent PNAS paper with Peter Doras that came out about 3 weeks ago. One thing that was fascinating is that they didn't shower for 3 days before they did the sampling, and yet you still see this incredibly dominant contribution of the beauty products.
That's really fascinating and begins to have new insight into how people differ. Would you like to comment on the studies recently done in the New York City subways where they actually found some pathogens, but as a normal part of the flora?
Yes, I'd love to comment on that. So actually, they primarily misidentified pathogens, and not just pathogens, but also agricultural pests. Where had those organisms really been present in the United States, it would have had really important implications for a number of trade treaties, right, like non-tariff barriers under the WTO and so forth. So you have to be very, very careful in doing these analyses if you actually want to identify organisms rather than finding community level differences. And so essentially the issue is that just because you find a gene that you can map to a pathogen reference genome, that really doesn't mean that it might not map to some other genome of something that's also benign, and it also doesn't mean that you necessarily have a complete reference database. So maybe it would map even better to something that's completely harmless that you just don't have in the reference database. And the lead author of that paper actually did a fairly detailed post on MicrobeNet where they reanalyzed a couple of their pathogen results and basically showed that the pathogen results are not well supported when you try to look at how much of the genome actually maps and are there alternative explanations for finding the pathogen. Now that said, in the Human Microbiome Project, where the sampling was a lot more rigorous, we did find a lot of evidence for genes that have only been found in pathogens where we were confident that the alleged pathogen was there. And these are things like Staph aureus and so forth, where we do know that those are at moderate prevalence in the body of even healthy people. And you can even take something like Mycobacterium tuberculosis, where most of the people who are infected with M. tuberculosis do not go on to progress to full-blown TB. So whether or not you get a disease is very dependent on host factors as well as microbial factors. But so it can be true that you have a pathogen and you're completely asymptomatic. So I guess typhoid malaria is the most famous example of that. But it's also true that it's very easy to accidentally detect pathogens that don't exist. So in one collaboration we had, the project was to find levels of salmonella in different parts of a crop. Different parts of a tomato crop, and through an initial analysis very similar to the analysis I used in the New York subway paper, our collaborators were very excited that they had found salmonella, and it was at different ratios in different parts of the tomato plant. But we were not able to confirm the salmonella using other molecular techniques. And when we looked at the metagenomic results, it was true that the metagenomic results had identified some salmonella, but at about the same frequency, they had also found reads from the duck-billed platypus, which I think was probably not on their experimental video. So yeah, so you have to be really careful about those identifications, especially if you don't have positive and negative controls that you're pretty sure are and aren't in the sample.
I'd like to share a question that's coming from our audience. How is sleep related with microbiome diversity?
Great question. So if you sleep more at night, you have a greater diversity in the microbiome, especially if you sleep fewer than 5 hours a night. That's relatively bad in terms of the overall association with diversity. And those low diversity communities have been associated with, I should turn that around. A number of different illnesses have been associated with low diversity communities, including obesity and inflammatory bowel disease, and several others. So one thing we are trying to figure out at the moment is whether sleep deprivation, in addition to giving you a less diverse microbiome, also changes it in specific ways that are related to those conditions, although we have not done that analysis yet. One other way that microbes are associated with sleep is that they're involved in circadian rhythm. So there was a group at the Weizmann Institute that did a very elegant study last year looking at jet lag, where essentially if you take a fecal sample from someone with jet lag, transplant it to a mouse, you disrupt that mouse's circadian rhythm, and it eats too much and develops insulin resistance. And fascinatingly, Jeff Gordon's lab and several others have shown that if you take mice that are germ-free, they don't have a circadian rhythm, and that circadian rhythm is only established once you colonize them with some microbes. So microbes can have a very profound connection to sleep in ways that we're just beginning to untangle, much like many of the other micro-brain connections that are only emerging now. And it's fascinating how rapidly this has come around, because as recently as 2010, basically everyone thought that the whole idea of looking for connections between the microbiome and the brain was totally crazy. It's just, just in 5 years, it's come around to being the sort of thing that, even your taxi driver claims that everyone knows.
That's great. Here's another question. Any follow-up on the impact antibacterial soap for the distribution of microbes, other than microbial, on the face or any part of the skin?
We haven't studied that at all. There's probably literature on it, but I don't know. I don't know who is working on it specifically. The skin mapping technology is very recent, and as a result, we're still trying to get IRB protocols approved to do interventions as opposed to observational studies, which are a lot easier to get approved. But that's certainly a major goal. One of the fascinating things about the skin is that there's a deep subsurface reservoir of microbes, and Rich Gallo here at UCSD has shown that they go all the way down into the dermis, which no one really expected that they would get that deep. Julie Segre at NHGRI has done some very elegant experiments on skin microbes, and what's fascinating is that her work shows that there's about 50,000 microbes per square centimetre on the surface of your skin and about 1 million per square centimetre subsurface. So on the one hand, what that means is that if you wipe the microbes off the surface of your skin, they get replenished very quickly from that reservoir below. But on the other hand, it implies that you might only have about 20 shots at wiping out that reservoir by wiping off the surface before it's essentially empty, and you might get recolonized by anything that's going around, including things that are going to be relatively harmful and really difficult to get out of the skin. So people who are in situations where they're sanitizing their skin very frequently, there's not a lot of data yet about what you get colonized with, but I think it's certainly an area that warrants substantial additional research.
Terrific. Here's another one. Do oral probiotics change or alter the GI microbiome?
Some of them do, some of them don't. The evidence is really mixed, and one thing to note is that even if they don't change either the species balance or the gene balance, they may change the gene expression of your indigenous microbes as they pass through. One thing to remember is that many commercially available oral probiotics have been explicitly selected not to establish in the gut for two reasons. So one reason is that it's easier from a regulatory perspective if you just say you're making a transitory change. The other reason is they probably want to keep selling you the probiotic every day, not just once. So a lot of them explicitly won't establish, but that doesn't mean that they won't have an effect as they're going through. And there are a number of clinical trials of oral probiotics that have demonstrated efficacy for specific probiotics and specific conditions. Just the same way that for drug trials, most drugs don't work on most conditions, but specific drugs are very effective for specific conditions. So you want to think about the probiotics much more like a drug, in that if there's a clinical trial that suggests that it works for a given indication, then it's worth trying. But that certainly doesn't mean that you should just take it as a routine thing.
So, Rob, I have a question on that area. Are you looking at the effects of chemotherapeutic agents on the biome, in the sense that both could they be modified by the biome and thus reduce their efficacy?
So we haven't done that, but a number of others have, especially like there's been a lot of work done on cyclophosphamide, for example, where its efficacy and toxicity are both linked to metabolism of that drug by the microbiome, and the same is true for a number of other chemotherapeutic agents. I think cisplatin there are some studies on, and there's also the very exciting potential of using as a drug target microbial genes rather than human genes, because you might be able to block specific aspects of microbial drug metabolism that would otherwise lead to toxic byproducts, or it could also lead to breakdown of the drug, thus clearing it from the body too quickly. So there's tremendous potential both for patient stratification based on the microbiome and for combination therapies where you get to modify the microbiome's ability to metabolize the drug. And this sort of thing is in large part why I moved to a medical school recently. It's very difficult to do that kind of thing long distance, whereas if you're right on site and you can talk to people who are actually doing the drug trials about what's exciting, it makes a huge difference to what you can potentially get done.
And I must mention that our program next week, which is the fourth in the series, will really address what new research, new data that's available on the impact of the microbiome on cancer therapy. Same time, same station. Here's another question. Regarding the skin microbiome, what work is being done on psoriasis? Namely, what is the change in the skin microbiome that might trigger the inflammation?
So several people, including Marty Blazer, have done work on that. That's one of those things where you really need to combine both time and space, which hasn't been done so much yet because the experiments relatively have been prohibitively expensive until very recently. But one of the characteristics of psoriasis is staph flare. Although what we don't know for sure is whether that's causing, whether it's a cause or an effect. One thing that's going to be increasingly important, as Julie Segre has shown in atopic dermatitis, is looking at fungi as well as bacteria. So in AD, which is also linked to staph flare, what you see is changes in both the bacterial and the fungal communities, and being able to integrate the bacteria, the fungi, the viruses on the surface of the skin, and for example, lysogenic phage repertoire, and the chemistry of the skin together, I think is going to be really essential for developing robust biomarkers that allow us to predict flare. I know a lot of other investigators are also working on psoriasis, know a lot more about it than I do. It's a very active research area at the moment, and these kinds of tools should be very useful for trying to untangle some of those questions.
Great. There's just one more that's been entered, but we do have another couple of minutes if anyone has additional questions. What is the state of the microbiome and obesity, particularly in the area of the brain-gut feedback loop? For example, has anyone performed brain scans and tracked 2 microbiome changes?
You've essentially described the next phase of our program project grant with Jeff, which we're just dealing with all the subcontract and consortium issues of transferring it from Boulder to UCSD at the moment. But yes, there's intense interest in that, both in the context of obesity and in the context of things like IBS, where you often have a visceral pain component. So, for example, Emran Mayer at UCLA has some very interesting coupled microbiome and brain scan data already. And then in the context of depression, where we're doing some very interesting work with Martin Paulus, who was at UCSD and is now at LIBR. So I think it's fair to say that the techniques for linking up the imaging data, where you have, again, very complex both spatial and temporal series, and the microbiome data, are just beginning to emerge at the moment. But it's certainly a very active research topic where there's a lot of excitement. One thing we do know is that there are many mechanisms for connecting microbes to obesity, everything from increased energy harvest to actually changing the behavior of the host. And one thing that's happened so far is that there's been relatively few integrative studies where you're looking at things like, like peptide hormones associated with obesity, and you're looking at immunology, and you're looking at microbiology, and you're looking at behavior, and you're doing brain imaging, and you're doing imaging of the rest of the mouse for body composition and so forth. So, so the issue is that getting that many investigators with different interests into the same room and talking to each other is relatively hard, but it's also absolutely worth doing. And you'll be excited to hear that it is finally happening.
Terrific. Well, Dr. Young, did you have anything additional you'd like to ask or say?
Well, I asked Rob to once again mention how people might be able to participate in his study that where you're looking for more samples. I think the audience might like that information, and I congratulated him on a really fascinating talk, and this is cutting edge, and this is where the field is going for sure.
Well, thanks so much, Howard, and thanks again for the invitation to do this. If you Google American gut, you'll get all the information about it as like the top few links.
Well, thank you, Dr. Knight. Thank you, Dr. Young. I want to thank everyone who's attended and for all the really great questions. And we'll look forward to seeing you next week. Our website, biopharmaresearchcouncil.org, will take you to all the registrations. And I truly invite you to be in touch with me. I'm Joanne Geer, the Executive Director, and you'll find my email and phone number on the website. Almost every one of our programs comes because someone says, I'd like to talk about this, or I'd like to hear about this. So please feel free to be in touch. We are making plans for the coming year and for next year, and your thoughts would be very much appreciated. So thank you, everyone, and have a lovely day. Take care.